Hello everyone. In the last video, we have seen the principles of inheritance till sex determination. In this video, we'll continue with this chapter. So here we have mutation. Now, what do you mean by mutation? Mutation is any change in the sequence of the nucleotides in the DNA. Clear? Let us say this is the DNA we have. We have A, G, T, A, T, G, C, A, something like this. Okay. Now we have base pairs also, isn't it? Now, if this is a normal DNA, okay, if this is a normal sequence of the DNA, if there is any change in this sequence of the DNA, then we call it as mutation in the DNA. Now, this mutation could be due to loss or gain. Okay, it could be due to loss of the nucleotides or it could be due to gain of the nucleotides. For example, if it is loss, let us say here I have removed this. All right. If this is loss, also it will be mutated. If it is gain, gain can be of two types again. So here again you have to remember. Okay, so if it is gain, what happens? It could be a duplication or insertion. Now, what do you mean by insertion or duplication here? Insertion is simply any nucleotide after this. For example, in this sequence, if I'm inserting one more nucleotide, say it's C. Okay, so this is called insertion. I'm simply inserting a new nucleotide there. But what do you mean by duplication? Duplication is repetition of a single nucleotide. For example, we have AGT. Okay, instead of now here in the same old example, if I tell you, instead of writing a new example. So before I have inserted C now. Now here, if instead of C, I'm simply inserting A here. So what happened? A is being repeated twice. Okay, so this is called duplication. Am I clear? So mutation can be due to two types. It could be due to loss or gain of nucleotides. Loss is simply deletion of any of the nucleotides. Gain can be of two types. It could be simply insertion of any nucleotide or duplication. We have a nucleotide before. Again, the same nucleotide, if it is repeating, then we call it as duplication. Clear? Now, mutations again can be again of two types, point mutation and frame shift mutation. Now, this I'll explain with the help of example given in your textbook. Let us say we have a DNA which reads something like this, CAT, 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 like this, okay? So, this is the sequence of the DNA we have. Now, obviously, we have the base pair sequence also here, okay? Now, in point mutation, what happens? Here we see the change in only one base pair. Single base pair will be changed. So here, how is the reading? CAT, CAT, CAT. So we have four cats, isn't it? CAT is cat. So we have four cats here. Now, in point mutation, instead of A, if I change it to C. Okay, so it is like cat, CCT, cat, and cat. Isn't it? So this is called your point mutation, wherein we have changed only one nucleotide. We didn't add or delete anything. Okay, if there was A before, instead of A, now we have C. So that will be your point mutation. So change in a single base pair is called point mutation. Example, sickle cell anemia. We'll talk this later in genetic disorders. Clear? Next, we have something called frame shift mutation. What do you mean by frame shift mutation? Again, in the same example, now, you have to remember frame shift will be due to deletion or insertion. Okay. And what is frame here? Frame is nothing but the reading pattern. Initially, we have cat, 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 and cat. Now, what is the importance of this reading pattern? See, each triplet of the nucleotides forms our genetic codon, if you remember molecular basis of inheritance. Okay. So, each triplet of nucleotide forms one codon and one codon codes for one amino acid. So, CAT will, after transcription, it will form mRNA, and after mRNA, we get the amino acids during translation process. Now, if you are inserting some additional nucleotide or if you are deleting, let us say if I'm inserting one nucleotide here. Okay, so after this CAT, if I'm inserting, say, here. So what is the reading pattern now? It will be CAT first. Okay, this CAT will remain as it is. Then it will be CCA. Then comes your TCA. Then TCA. Like this it will go. Isn't it? So what has happened? Reading frame is changed. reading pattern is changed due to the addition of one nucleotide in this sequence. Similarly, if you delete, what happens? For example, in this CAT, I am deleting this A here. So what will be the pattern now? CAT, okay. Then comes your CTC, isn't it? I have removed this one. So it will be CTC, then comes your ATC, then will be AT. So entire reading frame is being changed. Yes or no? All right. So whenever the reading pattern or reading frame is changed due to insertion or deletion of the nucleotides in the sequence of the DNA, then we call it as frame shift mutation. Clear? So we have two types of mutations, point mutation and frame shift mutations. Next, we have something called mutagens. What do you mean by mutagen? These are the agents which cause the mutation. Example will be your ultraviolet rays. Clear? Next, we have something called pedigree analysis. Pedigree analysis I'll take later after genetic disorders, because once we complete genetic disorders, you'll get some idea what kind of genetic disorders we have. And accordingly, we will be trying to analyze how to analyze those diseases in a family. Clear? So first we'll take up the genetic disorders here. Now coming to genetic disorders, we have two types of genetic disorder. One is called Mendelian and the other is chromosomal. What is the difference here? Mendelian disorders are those disorders which follows laws of Mendel, the law of dominance, law of segregation, all those things. Okay, especially the law of dominance. So here what happens in Mendelian disorders, the chromosomes, okay, basically chromosomes, we have 46 chromosomes. These 46 chromosomes are in 23 pairs. All right. Now, out of these 23 pairs, the first 22 pairs are what? They are called autosomal chromosomes. Okay, those are called autosomal chromosomes. The first 22 pairs out of 23 are autosomal, and the 23rd pair, that means the last pair, is called the sex chromosome. Now, what is the difference between this sex chromosome and autosomal chromosomes? Sex chromosome, we know it will determine the sex of the person. That means if the 23rd chromosome is, change the color here. If the 23rd chromosome is XX, then the person will be female. If the 23rd chromosome is XY, then the person will be male. Clear? So based on the last pair of chromosome, that is 23rd chromosome, we can tell whether the person is male or female. But before that, 23rd chromosome, we have 22 pairs of chromosomes. That means we have 44 chromosomes. Now, what is the use of these 44 chromosomes? First and foremost point, they will not determine the sex. They will not tell the sex of the person. Then what is the use? They will tell the other characters. For example, hair color, eye color, skin color, height. Okay, all other characters apart from the sex of the individual will be determined by the remaining 22 pairs. Understood? So the first 22 pairs, we call it as autosomal chromosomes, and the last 23rd pair, we call it as sex chromosomes. Now, if any mutation, if any defect is seen in autosomes, that is autosomal chromosomes, we call them as autosomal diseases. Simple. What do you mean by autosomal diseases? If any defect is seen in the chromosomes, which chromosomes? Autosomal, that is first 22 pairs of chromosomes, that is 44 chromosomes. If you see any defect, defect means what? Mutation. Just now we have read mutation now. Loss or gain or insertion or deletion. All those things will cause your mutations. So if there is any change in the DNA of the chromosomes, that is specifically autosomes, then we call it as autosomal diseases. So Mendelian diseases are classified into autosomal diseases and sex-linked diseases. So sex-linked disease will be the last 23rd pair of the chromosome will be your sex-linked disease. Okay, it could be sex-linked or Y-linked. So here in your textbook, X-linked chromosomes they have given. Am I clear till now? Yes. Again, this autosomal as well as X-linked chromosome, both of them are of two types, dominant and recessive. Clear? X-linked are also two types, dominant and recessive. Am I clear? Now, what do you mean by dominant and recessive? Law of dominance we have read now. What do you mean by law of dominance? In a heterozygous pair of alleles, okay, in capital T and small t, that is heterozygous pair of alleles, the character will be determined by the dominant allele. So whichever allele is dominant, that allele will be determining the character. What is the character here? Tallness was the character of the pea plant. It is because of the capital T there. Understood? So here also, in dominant disorder or dominant autosomal disorders, the trait, that is the disease, will be due to in the heterozygous. So here you have to remember that dominant or recessive both will be applicable when it is in heterozygous condition. See, when it is homozygous, definitely they'll show the disorder. If it is dominant homozygous or recessive homozygous, okay. In both the cases, homozygous, they'll definitely show the disease. The problem comes with the heterozygous. So in heterozygous condition, let us say we have capital T and small t only. So in heterozygous condition, if this is the disorder chromosome, then it will show the disease. Clear? And in recessive trait, it is not going to show the disease. Am I clear? So this is about the autosomal dominant and recessive. The same thing goes with your X-linked chromosomes also. So here I have made a chart of Mendelian disorder classified into autosomal and X-linked. And here you can remember the examples. Dominant, we have myotonic dystrophy. Recessive, we have 4 diseases: sickle cell anemia, cystic fibrosis, phenylketonuria, and thalassemia. Clear? This is all about your Mendelian disorders. Next comes your chromosomal disorders. Now, what do you mean by chromosomal disorders? See, once again, we have 46 chromosomes. Okay. If the number of chromosomes is being increased or decreased, then it will cause the chromosome. Basically, it can be increase in number or it can be due to defect in the chromosomes also. Okay. If the chromosome is getting halved, I mean, usually the chromosome will be something like this, isn't it? If it is half or if some part of the chromosome is being misplaced, okay? If it is cut. So whatever defect is seen in the structure of the chromosome, also in the number of chromosomes, then we call it as chromosomal disorder. But in your textbook, whatever examples they have given, it is based on the number of chromosomes. For example, the number of chromosome will increase or decrease. Clear? So instead of 46, person can have 45 chromosomes or the person can have 47 chromosomes, like that. So number will be increased. Clear? Next, coming to the disorders. Now here I have written some points here. Now we have some terminologies like homozygous, heterozygous, and we have one more called hemizygous condition. Now, what do you mean by hemizygous condition? I'll tell you once I explain this color blindness. Okay, then you'll come to know. So color blindness, first and foremost, it is sex-linked recessive. See, autosomal dominant disorder, myotonic dystrophy, it is just mentioned in your textbook, but they haven't explained it anywhere in the textbook. Okay, so we'll not go in the explanation part. You have to just remember the example of autosomal dominant is myotonic dystrophy. Clear? Now, whatever diseases we are going to discuss, that would be your X-linked recessive disorder. Again, X-linked dominant disorders, examples are not mentioned in your textbook. We'll look at this table again. I'll show you. Okay. So here, Mendelian, autosomal X-linked, dominant myotonic dystrophy is mentioned. Clear? Then autosomal recessive examples are mentioned, and all the, for not all, cystic fibrosis is again not mentioned. Only 3 examples they have mentioned. I mean, the description is given. So cystic fibrosis again will not touch. Clear? Again, they have given the examples of X-linked from disorders, color blindness and hemophilia. So what are the things we're going to discuss briefly here is sickle cell anemia, phenylketonuria, thalassemia, color blindness, hemophilia, Turner's, Downs, and Klinefelter's. These are the diseases we are going to discuss. Okay. Cystic fibrosis, not mentioned in the textbook. Myotonic dystrophy, not mentioned. Apart from that, dominant X-linked disorder is also not mentioned. So we'll not touch those. Now, coming to the first one, color blindness. Now, what is this color blindness here? X-linked. recessive disorder. Okay. So first we are going to discuss about the X-linked recessive disorders, color blindness. Now here, first point, sex-linked or X-linked, okay, recessive disorder. Now what happens in color blindness is person is not able to differentiate between the red and green color. Why the person is not able to differentiate? Because there is defect in the cones, red and green cones of the eye. We have cones in the retina, I mean, in the eye, in the retina, yes. The innermost part of the eye, we have cones, isn't it? Rods and cones, if you remember. If you have done the eye is deleted, I know. But if you have read that, so you might be knowing that we have something called cones and rods in our retina part of the eye. And if out of that cones, if red and green cones are getting defective, then the person is not able to differentiate between the red and green color. And that is called color blindness. It is frequent in males compared to females. Very, very rarely the females are affected, 0.4%. You can imagine that is very less. Okay. In males also it is very less, 8% of the males. Now, here, what happened? Now we have conditions. So these are the things they will ask in the exam. So first condition is like, okay. So here we have female with the color blindness, okay? And here it is male, which is normal. So female here, sorry, this is not color blindness, it is recessive, right? X-linked recessive. Now, since it is heterozygous female, okay, XC, X is heterozygous. Let me write down here. Heterozygous female. Now, since it is recessive, okay, what happens? Is the female affected or not? Female is a carrier. Female is not affected. That means phenotypically, morphologically or phenotypically, the female can easily differentiate the colors. Okay, she is not affected. But genetically, she is affected. So that means it is carrier. She is a carrier. Whereas male is a normal here. Okay. So what happens to their progeny? Out of 4, okay, we get 2 females and 2 males here, 2 daughters and 2 sons. Out of that, 1 daughter will be carrier. Clear? 1 daughter will be normal. I mean, both the daughters will be normal, but one will be carrier, the other is completely normal. And in son also, 1 son will be affected and the other son is not affected. Now, why this son is affected? See, here the female is not affected, I'm telling. I'm telling female is carrier, but coming to the son, even though only 1X is affected, I'm telling that son is affected by the, disease. What is the reason there? In males, we have only 1X chromosome, isn't it? Male is X and Y. In this, we have only 1X chromosome, and to that X, it is defective. So here male will be defective. And please remember this condition where a recessive character, a recessive disorder is present on the X chromosome, we call it as hemizygous condition. Clear? So this is hemizygous condition. So this is the first condition of color blindness, wherein the female is a carrier, male is normal. So you have to remember the progeny, what happens to the progeny. You need not by heart, you have to just remember the concept, what happens here. So you should be able to make a cross, how to make cross. Okay, I'll tell you how to make by giving the second example. So we'll see the second example wherein the female is defective. That means both the X chromosomes are carrying the defective gene. Clear? So here the female is affected by color blindness and crossed with a normal male. Male is normal. Now what happens? This X goes with this, XCX. This X goes with this. All right. This goes with this. This is how you have to do in the exam. And this again goes like this. Clear? So how is the progeny? Let us check now. Now this is female and this is female. So what is happening to females? Females, both of them are carriers. Okay. Coming to males. What happened to male? One male is normal. The other male is affected. So what is the difference here in the first case and the second case? In the first case, 1 daughter was carrier, the other daughter was normal. Isn't it? This daughter is normal. Whereas in this case, both the daughters are carriers. Clear? This is the difference here. Now I can have the third condition also, wherein female is normal but male is affected, like this. What happens in this case? Okay, when it is crossed, now this X goes with this XC. Again, this goes with Y. Okay, now this X goes with this XC. And again, this goes with. Now what happens here? Let us analyze. Now this is female. Here also we have female. So what happens to female? Both the females are what? Carriers. Both the males are what? Normal. Okay, so these are the three conditions they can ask you in the exam. Simply what they can give you is how the questions are framed. I'll tell you. They'll tell you the condition of the progeny like this. They say that out of 4 children, 2 females were carrier and 2 males were normal. So 50% of the females were carrier and 50% males were normal. So what was the condition of the parents? This will be the answer, question. Okay. So what is the condition of parents? Male is affected, female is unaffected. Clear? Similarly, they can ask you like out of 4 children, 1 son is affected, or we can say 50% of the son is affected and the remaining 50% is normal. But both the females are carriers. Okay. So answer will be again, female is affected, male is unaffected. So this is how they frame the questions based on X-linked disorders. The same thing goes with hemophilia also. Clear? Next, coming to hemophilia, again, this is also a sex-linked recessive disorder. It is also called royal disease, not because of the royalness of the disease, okay, because it was first observed in Queen Victoria's family, and since their family is royal family, so this disorder is called royal's disease, okay. And here, the female becoming hemophilic is very extremely rare because here also males are more prone to disorder compared to female, and whenever the male is affected, by the time the male attains maturity or by the time they attain the reproductive age, they will be dead, because what happens here is, even though the male is affected by a slight disorder, like slight injury, slight cut on their skin, they will be profusely bleeding and that will lead to death. So males are unviable. That means they cannot survive for many years. As a result, whichever male is affected, male will be dead. Clear? So females becoming hemophilic is extremely rare in this case. The same thing, now the cross, if you see the same, it is same to that of your color blindness. I'm not repeating here again. So there will be 3 conditions. What will be the conditions here? Female carrier, male affected, male normal, female affected, male normal, female normal, male affected. Okay, these will be the 3 conditions for hemophilia. So this is about your X-linked recessive disorders. Next, we'll talk about sickle cell anemia, which is autosomal recessive. Okay, it is autosomal recessive disorder, and we call it as qualitative disorder. Why it is called qualitative, I'll tell you later. So what happens basically here is, if you remember, we have done sickle cell. I have mentioned sickle cell anemia somewhere before in this class. Yes, can you tell me where? Point mutation. Isn't it? In point mutation, I have mentioned sickle cell anemia. So what do you mean by point mutation? Just now we have read now. What happens in point mutation? In the sequence of nucleotides in the DNA, one of the nucleotide will be changed, isn't it? Say we have A, G, A, C, U, T like this. So one nucleotide will be changed to some other. Same thing happens in sickle cell anemia. So basically what happens here? We have in this sequence, for example, we have something like G, U, A, C, A, something. Okay, this is something. And then comes your G, A, G like this. Okay. Now, instead of this G, A, G, what happens here? This is mRNA. This is in mRNA. Clear? We don't have U in DNA, right? So this is an mRNA. And instead of A, G, A, G, we have what? G, U, G. And because of this G, U, G, Before GAG was coding for glutamic acid. Now the GUG will be coding for valine. Translation. Clear? And where do we see this? In the sixth place of the amino acid. Say this is your mRNA. We have triplets of codons like this. 3, 4, 5, 6. Okay, so this is the sixth position of 3 nucleotides. That means codons, I'm telling you. So instead of GAG, if it is GUG, then we should actually have glutamic acid at the sixth position. But since from GAG, GAG, it is modified to GUG. Remember, like GAG to GUG. Okay. So since it is modified to GUG, glutamic acid is modified to valine. And because of the presence of valine in the sixth position, what happens? The RBCs which are present in our blood, okay, they cannot undergo polymerization. What do you mean by polymerization? They cannot undergo, maturation. So RBCs are not able to undergo maturation because of the low oxygen tension inside the blood. Okay, they are not able to carry the oxygen properly. And because of that, instead of having a circular shape, the RBC will attain a sickle cell. They are not able to grow properly. You can just simply remember in simple terms. Because of the sixth position, valine in the sixth position, they are not able to get sufficient oxygen. Okay, as a result, the RBC is not able to grow properly and they will remain in the sickle shaped structure. Okay, so this is about the sickle cell anemia. Now, I told sickle cell anemia is a qualitative disorder. What do you mean by quality? Here, the quality of the hemoglobin is being changed. See, what happens here? Basically, where is this valine and everything? We have RBC. Let us say this is my RBC. Okay. And inside this RBC, let me draw the RBC something like this. Why did I draw like this? RBC is biconcave, isn't it? And in this RBC, I have hemoglobin. We have lots and lots of hemoglobin, isn't it? If I take hemoglobin out, now what is this hemoglobin made up of? It is heme plus globin. All right. Now what is this heme again? Heme is made up of iron plus porphyrin group. Okay, so heme is made up of a molecule of iron and porphyrin. Let me draw the structure and not draw completely. Let us say this is iron, and to this I have the porphyrin group, something like this. So we have 4 porphyrins. Okay, to each heme. Like that, we have 4 heme in one hemoglobin. One hemoglobin molecule, if you take, we have 4 heme molecule and 4 globin molecule. Okay, I'm telling you hemoglobin structure because this will be helpful for you to understand both sickle cell anemia and thalassemia. In thalassemia also, we have to talk about hemoglobin. Clear? So please try to understand the structure of hemoglobin here. Hemoglobin, one molecule of hemoglobin is made up of 4 molecules of heme and 4 molecules of globin. So here I have drawn one structure, one molecule of heme. Heme instead is made up of 1 iron and 4 porphyrins. So this is the structure of one heme molecule. Like this, we have 4 heme molecules in the hemoglobin. Am I clear? And it has got 4 globin structures. 4 globin. Now, globin is what? The protein. So basically, the protein which I was talking in the sickle cell anemia is the protein of globin. Clear? And this globin is of 2 types. What are they? We have alpha globin and beta globins. So, out of 4 globin chains, we have 2 alpha and 2 beta. Clear? Now, which globin chain is being affected in sickle cell anemia? It is beta chains of the globin. Please remember that. So, roughly, if I want to draw the structure of hemoglobin, where should I draw? So let us assume this is my iron porphyrin like this. Okay. So I have 4 heme molecule, something like this, isn't it? 2. Then again, are you able to follow? Okay. So these are my 4 heme molecules. Okay. And to this 4 heme molecules, I have iron and porphyrins. Clear? And to this heme molecules, I have 2 globin chains. Say this is 1, 2, and 3 and 4. Okay. So here, let me say this is alpha globin. Both of them are alpha globins, and these are my beta globins. Clear? So this will be your 1 molecule of hemoglobin. Now, in sickle cell anemia, what is happening? This chain of protein, this chain of amino acids is being affected. So quality of this globin chain is getting changed, isn't it? Quality is changing here. Hence, this is called qualitative disorder. You'll get some more clear idea when I talk about thalassemia. So first, let us take thalassemia, and then again, I'll come to this point, qualitative disorder. Now, if you talk about thalassemia, what is happening in thalassemia? It is actually a quantitative disorder. Clear? So, quantity of hemoglobin is affected here. How? First of all, thalassemia is of 2 types, alpha thalassemia and beta. Now, why alpha and beta? It is based on the type of globin, alpha globin and beta globin. If alpha globin chain is affected, then we call it as alpha thalassemia. If beta globin chain is affected, then we say beta thalassemia. Okay. Now, what happens here is alpha thalassemia. Okay, please try to understand here again. Alpha thalassemia, I told alpha thalassemia is due to the effect, the disorder in alpha globin chains. Now, if you talk about the alpha globin chains, alpha globin chain is nothing but a protein, isn't it? And how do we get this protein? Due to a gene. Yes, gene expression leads to the formation of protein. Now, where exactly this gene is present? This gene is present in chromosome number 16. Okay. So the gene responsible for the synthesis of alpha globin chain is present in chromosome number 16. So if I draw the structure of chromosome 16, okay, this is homologous pair of chromosome 16. Now here I have basically 4 alleles. Okay, one is hemoglobin A1, hemoglobin A1. So this is one gene with 2 pairs, 1 pair of alleles. Similarly, I have hemoglobin A2 and hemoglobin A2. So what is happening in chromosome number 16, okay, whether it is male or female, in chromosome number 16, we have 4 alleles responsible for the synthesis of alpha globin chain. Clear? Even if one of the chain is effective, for example, this gene is defective. Which one? Let me take this one. Okay, this gene is defective. This pink one is defective. Now, can this gene synthesize the protein responsible for it? Can it express the protein? No, isn't it? So what happens for the synthesis of this alpha globin? Only these 3 genes will be responsible. Okay, whichever part of the alpha globin was supposed to be synthesized by this gene will not be synthesized. So what is happening? Let us take this example. Say this is the chain of the alpha globin. I am dividing it into 4 parts. Okay. This was from this gene, second, third, and fourth. So all these 4 parts, okay, this part, this one, this, and this are synthesized by the 4 different alleles. If one of the allele is getting defective, what happens? That part of the globin chain will not be synthesized. This part will not be synthesized. Similarly, if this part is defective, okay, now this part will not be synthesized. Only from here to here only this alpha globin chain will be synthesized. Are you able to follow me? Yes? So hence we call thalassemia as quantitative disorder because here the quantity of the alpha globin chain is getting reduced. If this gene is defective, this part will not be synthesized. Okay. If this gene is defective, this part will not be synthesized. Similarly, if this is defective, this part will not be synthesized at all. If this is defective, overall, entire alpha globin chain will not be synthesized. Okay. Hence, we call it as quantitative. I hope you are able to understand. Okay. Now, coming to beta thalassemia, the gene responsible for beta globin synthesis is present on chromosome number 11. And here we have only one gene. Okay, the difference you have to remember. For alpha globin, we have 4 alleles or 2 genes responsible for that. Whereas in chromosome number 11, for example, if this is the chromosome number 11, okay, only one gene is responsible for the synthesis of beta hemoglobin, that is hemoglobin B and hemoglobin B here. Clear? If one of the gene is defective, if suppose this is the chain of the beta globin, half of the globin chain will be not synthesized. Similarly, if the other is defective, this part will not be synthesized. Okay. So hence, thalassemia, we call it as a quantitative disorder. And again, if you come to this sickle cell anemia, I said it is qualitative. Entire alpha and beta globin chains will be synthesized, but the quality is not good there because instead of glutamic acid, we have what? Valine. I hope I'm clear. Okay. So this is about sickle cell and thalassemia. Next, we'll talk about something called aneuploidy. Aneuploidy is a gain or loss of entire chromosome. It is not one nucleotide or 2 nucleotides, entire chromosomes. For example, excuse me. Say we have 46 chromosomes. Okay. Out of this 46 chromosomes, if I have lost or gained, if I have 45 chromosomes or 47 chromosomes or 48 chromosomes, then that condition I call it as aneuploidy. Okay. So instead of 46, I will have 45 or 44 or 47 or 48. 46 will not be there in case of aneuploidy. Clear? Next, coming to polyploidy, here the set of chromosome will be increased. We have diploid set, that means we have 46 chromosomes. We get 46 because of 23 pairs, isn't it? But in polyploidy, what happens? Instead of 23 pairs, instead of having 2 in this, the entire set will be doubled. So instead of 2n diploid, we will have 3n or 4n or 5n. Okay, so if this is the condition seen, the entire set of chromosomes is being increased. So 46 plus 23, how much? 46. If it is 3n, 3n will be 46 plus 23. No? So what will be the answer? 6, 7, 8, 9. Then 4, 5, 6. 69. Correct? 4, 5, 6. So if 69 chromosomes are there, it is not called aneuploidy, it will be polyploidy. And polyploidy, please remember, it is more common in plants, not in animals. So if entire set of chromosomes is increased from diploid to triploid, tetraploid, hexaploid like that, then it is called polyploidy. But instead of increasing the set of chromosome, if only 1 or 2 chromosomes are being increased or decreased, then it will be aneuploidy. Now coming to the examples of aneuploidy, first we have Down's syndrome. Down syndrome is autosomal disorder. What do you mean by autosomal? In the first 23 pairs, I told you, isn't it? First 23 pairs, if any one chromosome is increased. So basically here, usually, if you consider 21st chromosome, in 21st chromosome, usually we'll have one pair like this. One from paternal, other from maternal. One from father, the other from mother. Okay. If this is a condition, fine, it is good. Okay. But due to some problems during meiosis, if you have one more chromosome here, Okay, so instead of one pair, we have triplet of chromosomes, which is forming our 21st chromosome. Then that condition is called as Down's syndrome. And please remember, Down syndrome can be in any gender because it is autosomal. 21st chromosome is what? It is autosomal. So it can be seen both in males or females. Clear? So we call it as trisomy of 21st chromosome. Why trisomy? Because instead of pair, we have three chromosomes, which is present in the 21st chromosome. So additional copy of chromosome, that is 21st chromosome, hence it is trisomy. And it was first described by Langdon Down, hence it is called Down syndrome in 1866. What are the conditions we see here? Short stature of the person, furrowed tongue, mental retardation, palm crease. These are the things we see in that particular children. Clear? So Down syndrome is seen in any gender, that is autosomal. Next, whatever we are discussing, that is Klinefelter's and Turner's, is related to sex chromosomes. But all the three are aneuploidy. Please remember, okay? Klinefelter's, usually 46 chromosomes we have, okay? And in 23rd chromosome, we have, if it is a male will have X and Y chromosome, isn't it? But instead of X and Y, if the male has got additional X chromosome in him, then the condition is called Klinefelter's syndrome. Okay, so what will be the number of chromosomes now? It will be 47 now. Usually it is 46. But due to additional X chromosome in males, we get that condition called Klinefelter's. Okay, so condition will be 47 now. So 47 chromosomes, males with XXY. Now the person will be male. Appearing wise, person will be male. So overall masculine development, however, gynecomastia is seen, but the person will be sterile. So that means they cannot undergo reproduction. Okay. Next comes your Turner syndrome. Turner syndrome is seen in females. Females again, 23rd chromosome. If you see, we have X and X. Okay, so two X chromosomes should be present in female. But due to some condition, if we lose this chromosome, okay, if the second X is lost, so we have what called XO there, isn't it? We call it as XO. So what will be the number of the chromosomes now? Instead of 46, we will have 45 chromosomes, isn't it? So 44 are the autosomes here, autosomal chromosomes plus XO. Only one X chromosome is present. That condition is Turner's, which is seen in females. So total number of chromosomes will be 45. Female with only one X chromosome, person will be sterile here. Rudimentary ovaries, okay, no functional ovaries, no secondary sexual characters, person is sterile. Okay, so this is all about the genetic disorders. Now we'll go with the pedigree analysis, which I have left. So if you talk about pedigree analysis, now you will be able to understand properly. So for pedigree analysis, now what do you mean by this? Actually, it is analysis of inheritance of traits in several generations of a family. Inheritance means transfer of traits or characters. It can be characters or diseases, anything. If we want to study any particular transfer or inheritance of disease or character in generations, I mean in a family tree, then we do something called pedigree analysis. Okay. For this, we have certain signs like square is male, circle is female, all these things, please go through. Next, we have something called clues. So before clues, I'll tell you the trick to solve the pedigree analysis, very useful for exam. Okay, so basically from this topic, what they ask is, they'll ask 6 diseases. Okay, they'll ask only 4 diseases, but sometimes they ask the other 2 also in the options, so you should not get panic here. What are those? First will be your autosomal dominant. All this just now we have discussed, isn't it? Then we have autosomal recessive. Can you tell me the examples? X-linked dominant, X-linked recessive. Okay, these we have just now done. Autosomal dominant, myotonic dystrophy, autosomal recessive, or what? Yes, just now we have done. You should tell me. Thalassemia, sickle cell anemia. X-linked dominant, examples are not given. X-linked recessive is what? Yes, please write in the comment section. Okay, all right. So these are the things we have done. Now, the fifth is cytoplasmic inheritance and Y-linked disorders. Okay. So these are the 6 disorders they usually ask in this pedigree analysis concept. So you have to check the options first. If any pedigree chart comes in the exam, first and foremost, you have to go through the options. Before reading the question, you first go through the options. Okay. If these are the 4 options they have given, then read the question. And then you have to ask 3 questions to yourself. What are those? Okay, please try to follow this trick. It will be very useful for you to solve the pedigree analysis charts. Okay, whatever chart comes, if you apply this trick, 100% you'll get the answer. But you have to do it carefully. Okay, so what did I say? First, you have to go with the first, as when you see that chart, pedigree chart, first you see the options. If you have these 4 in the options, then you have to read the question properly, and then you have to ask whether, okay, we have generations there. Okay, so I'll give you one example. Let me take one example. Randomly, I don't know. Okay, let me take one example. Okay. This. Okay, let us say this is the exam. Okay, right. So by using this example, now we'll try to solve the pedigree chart. Say this is a random chart I have taken. Okay. Now, what is the first question you're going to ask? Are all the affected children have at least one affected parent? What do you mean by this? First, you have to look at the generation. First, you have to see which generations. Okay, these are the parents we have. Okay, these are the parents. Then this will be your first generation. Okay, this all will be first generation. This will be your second generation. And this will be your third generation. Clear? So you have to check the children of all the generations, and then you have to see whether their parents are affected or not. Affected children, if their parents are affected. Let me take an example here. Now, first generation children I'm seeing here. Okay, is any child affected? Yes, this child is affected. Is the parent affected? Yes, one of the parent is affected. So answer is yes for one generation. Let me check for the second generation. In the second generation, if you see it carefully, none of the child is affected. This is third person, isn't it? He is marrying her. But if you look at the children here, none of the child is affected and their parent is affected. Okay. Now this is not applicable because you have to check only for the affected children here. Affected children only you have to see. So this is not applicable. Clear? Now go with the third generation. Child is affected and one of the parent is also affected. Clear? So what is the answer for the first question we have? Answer is yes. All affected children have at least one affected parent. First generation, child is affected, parent is affected. Second generation, no child is affected. So we'll not check for the second generation. Go with the third generation, child is affected, one of the parent is affected. So answer is yes. If the answer is yes, then the condition is a dominant disorder. Okay. If the answer was no, now we don't have no, so I'll not go with the no now. Answer is yes in our case. Clear? Now, if the answer is yes, what is the next question you are going to ask? First you have to look at the males. Remember, dominant males, recessive female. Like that you remember, okay? Dominant males, recessive female. So if the answer is yes, the condition is dominant. And if it is dominant, you have to look for the males. What you are going to check there? Affected males, if they have whether the affected males have affected mothers and affected daughters. Then again, yes or no. Okay. So let me erase this again. So in the first time we got answer as yes, isn't it? Oh, oh, I lost her. Oh my God. Something like this. Mhm. So this was a condition, isn't it? Now, correct only, no? Hmm. Now, check for the males. So here, parent male, are all the daughters affected? Let us check. This daughter is not affected, but this daughter is affected. So answer is no. The first case itself, it is no. Okay, similarly, I'll go with the next one. This male is affected. Okay. If one of the daughter is affected, the other daughter is not affected. Clear? And that's it. So answer is no here. So answer is no. And since the answer is no, answer would be autosomal dominant. This is how you have to check. So please remember, whenever you are doing, you have to check for each and every generation. For pedigree analysis, I'll again make a detailed video, okay, for 10 to 15 minutes, wherein I'll include all the different types, examples, proper examples, and then I'll try to solve them by using this trick. That would be helpful, okay? For now, just you practice by using this two or three questions, and then definitely you'll get the answer. And one more point I just wanted to tell you is, in your textbook, they have given myotonic dystrophy, example of autosomal dominant. For that, they have given a pedigree chart, and if you apply this trick to that chart, it won't work out. Okay, so I'm telling you that also. So this trick will not work out for myotonic dystrophy. So what you will do now? Please by heart that. Okay, by heart that chart. So when you see that chart as it is in the exam, you should be able to identify that, yes, that chart is myotonic dystrophy. Like that, you have to by heart. I'm asking you to by heart only one chart. Clear? So please do that. And remaining any chart if they give in the exam, 100% you'll get the answer by using this trick. That is my guarantee. Okay. So this is the trick for solving the pedigree analysis, pedigree charts. And as I have told, the 4 diseases we can do by this trick. Okay. And I have told you the other 2, cytoplasmic inheritance and Y-linked disorder. What are they now? Mitochondrial inheritance or cytoplasmic or maternal inheritance is nothing but a condition where the cytoplasm of the female is affected. Okay. So when the cytoplasm of the female is affected, we know during the fusion, that is fertilization, the nucleus from the sperm will enter into the cell, that is the egg cell of the female, and inside we have the nucleus where the fertilization takes place. Now, whatever zygote we get, only one zygote we get here. Okay, so whatever zygote we get, basically this zygote, the cell of the zygote is from the egg of the female. So if the cytoplasm of the female is defective, obviously that defect will be seen in the zygote. And if the zygote is defective, obviously the entire person will be defective. Understood? So here, whenever, if the females are affected, now in the pedigree chart, how will you determine this mitochondrial inheritance? See, mitochondria is present in the cytoplasm. Clear? So mitochondrial inheritance or cytoplasmic or maternal inheritance. If any female is effective, then if all the children, let me take the example, you'll get confused unnecessarily. Let me take here. Say we have a condition where female is effective. Okay. And here, say we have all this generation. And again, okay. And let me write here one more. Okay. Hmm. All right, then this. Okay, this is the condition. So what is there? See, female is affected. All the progeny, how many? 1, 2, 3, 4, 5. All the progeny of this female will be affected. Clear? Next, here female is effective. And again, if you see the progeny, all the progeny are affected. Okay. So when female is affected and all the progeny, without any exception, are affected, then that condition we call it as mitochondrial or cytoplasmic inheritance. Okay. So here, male, even though the male is affected, suppose this male or this male, this affected male is marrying to a female. Now, why these children are not affected? Because the the nucleus of this male will enter into the egg of this female. So, cytoplasm of the female is normal, is that right? So, children will be normal here. I hope you have understood. Clear? Next, coming to Y-linked disorder. In Y-linked disorder, in the pedigree chart, if all the if all the males in the chart are affected, then it is Y-linked. All the males. Clear? All right. So this is all about the pedigree analysis and the disorders. And the last point I just wanted to mention, I've just got the formula chart here, which is useful for this chapter. Like we have types of gametes. In order to calculate all these things, types of gametes and everything, you just note down this formula. I'll make a separate video by taking certain examples. Okay, so we'll use this formula and a particular example, because random examples I don't want to take. I'll take some valid examples and then we'll try to solve those examples by using this formula. Okay, so this is all about the chapter Principles of Inheritance and variations. Okay, so please try to share this video with your friends, and thank you.